Hi everyone, this is Tuval from Element Biosciences, and we're going to spend the next 5 to 10 minutes learning about how LoopSeq long-read sequencing can generate value for microbiome sequencing, and more specifically, long-read 16S sequencing and its use in microbiome characterization. So let's get started. Just as an overview, LoopSeq is a technology that leverages short-read sequencing infrastructure in order to generate highly accurate long reads on any short-read sequencer. For those of you that are interested in learning more about how LoopSeq works, there's an overview webinar that you can watch that takes a deeper dive into how LoopSeq chemistry and software actually generates long reads from short reads. Okay, so let's get started. What we'll do today is we will go over 3 different publications from our customers that did various types of comparative analyses between LoopSeq long-read 16S sequencing and other types of long-read sequencing and short-read sequencing. We'll go over those results and see what they found. So let's start with this scientific reports publication from 2021. What this group did is compare V3, V4 sequencing on an Illumina sequencer using a MiSeq with paired N300 sequencing to LoopSeq V1, V9, 1500 base pair sequencing. So this is the V3, V4 segment that was sequenced with these 2 primers, and then the LoopSeq amplified the V1, V9 using the 27F1492 primers to amplify V1, V9, 1500 base pair fragment. And this just goes to show that the fragments were had UMIs distributed throughout their lengths, and then we used our consensus-based assembly process to make synthetic long reads that contained all 9 parallel regions on each read. So those are the 2 technologies that were compared here. And now let's move on to what they found. So first of all, in terms of representing diversity, they did 2 different types of analyses here on the exact same samples. So this analysis shown here in panel A shows the number of observed OTUs. OTUs are essentially, or they're also known as ASVs in the microbiome space. ASV stands for amplicon sequence variants. So these are essentially unique 16S reads, right? So they're counting the unique number of sequences that they find in the data, basically taking all the reads, clustering them at 100% homology, and then counting the number of ASVs or unique reads. So you can see that full-length 16S detects more unique sequences in a microbiome sample compared to short-read sequencing, despite the fact that it's actually sequenced to a more shallower depth. And then the remaining figures here show various types of alpha diversity metrics like Shannon and Simpson. And in all of them, you can see that the diversity of full-length sequencing, which is the right bar here, is higher than the diversity of short-read sequencing shown in this V3, V4 box. Moving on to the next figure of the same paper, the authors show the number of unique taxonomies detected at different types of taxonomical resolution, starting from the phylum level all the way down to species level. And really, there aren't major differences between them. The overlap is more or less 100% until you get down to species level, when they start to kind of depart from each other. And you can see that at really the difference becomes more clear as the taxonomy gets more resolved. So when you get down to species level, you can see that full-length 16S sequencing, shown in blue here, detects more taxonomies at the species level compared to V3, V4 sequencing. And that's really probably the greatest advantage of long-read 16S sequencing compared to short-range 16S sequencing, is that you have much more resolution at the species and sometimes even strain level, which we'll show in the next few slides. Moving on to, there's 2 more figures in this paper. So what they're showing here is essentially what they showed in the previous figure, just presenting it in a different way. They're showing that in terms of relative abundance at the phylum level, both methods look the same, V3, V4, and V1, V9. Then when you go down to genus level, you start seeing differences, but really the differences more pronounced here at the species level. You can see that this is full-length 16S sequencing and this is V3, V4. So it's pretty obvious here that when you look at the 16S full-length sequencing, that there are way more species detected here than here. So the resolution here is much greater. We're detecting way more species here than we are here. And reason being that just V3, V4 alone does not always contain enough taxonomy information to resolve down to the species level. Last figure from this paper shows this violin plot where, again, what we're doing here is we're showing the percent mapping with a database. I think they use the Silva 138 database here, or sorry, an NCBI database. And what you can see here is that when you go down to species level, which is the figure on the right here, you see that there's a big chunk of the data for LoopSeq here that is highly resolved, close to 100%. And that's where most of the data is concentrated at the species level, like with very high confidence, close to 100%, which is not so much the case for V3, V4 sequencing. Okay, moving on to another paper published in bioRxiv in 2020. What we're seeing here is the ability of full-length long-read sequencing of 16S to resolve strains, not only species. And the way that strains were resolved in this paper is very interesting. So what they're doing here is sequencing a single strain and showing how long-read sequencing can resolve copy number variation of 16S in the bacterial genome. So 16S can exist in one or more copies in bacterial genomes, typically more than one copy. It can be anywhere up to 10 different copies, sometimes even more. So 16S sequencing provides not only more resolution because you're reading more variable regions, but also you can differentiate. There's enough information in that long read in order to differentiate between the different copies that are present in the genome, which is not always the case with the short reads, right? Because if the variation from one copy in the genome to another copy in the genome exists in a variable region that you're not sequencing, for example, it exists in variable region 9, but you're only sequencing V3, V4, then you really won't be able to differentiate between the different copies in the genome. So what the authors did here is they mapped all the different copies. If I'm not mistaken, there were 10 copies in this Bacillus subtilis genome. So there were 5 copies that were identical, and then 2 copies of a different 16S, and then 3 copies that were unique. So it's 5, 2, 1, 1, 1, altogether 10 different copies. And when you sequence that strain with short read sequencing, which is shown here, you can see that in the blue bars, you can see that you're not really getting the accurate representation of the 16S copy number in the genome. The expected versus loop are the red and green bars here. So you can see that we're actually on the y-axis plotting the copy number variation properly, whereas with short read sequencing, for several of the copies, right, this one here and these 2 here, for 3 out of the 5 unique copies, they're actually not accurately accounting for the copy number. And this, you can think about this as sort of a signature for strains, right? How many copies they have in the genome and what is the copy number for each one of the unique sequences in the genome is a strain level identifier in microbiome sequencing. And this is something that is enabled, this property is enabled by long read sequencing, and it's almost impossible to detect using short read sequencing. Okay, the last paper that we'll go over was published in Microbiome in 2021. In this paper, the authors did a comparative analysis between LoopSeq, PacBio, and Oxford Nanopore. In this specific analysis, they're comparing a complex microbiome sample sequenced only with LoopSeq and PacBio with 2 different generations of their CCS reads. And basically what this figure shows is that the diversity in these samples is captured almost identically in all these 3 different technologies. So that's just to show that LoopSeq is, I guess, compatible or just in accordance with the existing long-read sequencing technologies. Another figure from the same paper did a comparative analysis, this time for error rate in over all 3 different long-read sequencing technologies, LoopSeq, ONT, and PacBio, CCS reads, and they sequenced full-length 16S molecules, which are these data points here. Full-length 18S, ITS1, and ITS2 molecules. These are roughly 2,500 base pairs. See, those are data points here. And then full ribosomal RNA clusters, which are 5 kb in length, and these are the data points here. And what they're plotting is the fraction of error-free reads, so these are reads with 0 errors in them, as a function of the length of the read. You can see that essentially all the different read lengths, LoopSeq has a much higher fraction of error-free reads, which means that you have way less false positive results in your data. For those of you that are interested to learn more about our products and our services, please visit us at the Learn section of our Loop Genomics website and at elementbiosciences.com. Thank you very much for your attention.